All right, I got my taskbar hidden. It didn't even dawn on me that it was. Anyway, don't ask. Let's not get into the technicalities of why the taskbar was there. But so you can see this diagram now. I think we've seen this before. But anyway, now we can see all the entire screen now. All right. So this is glycolysis. This is where we take glucose, we generate 2 pyruvates, and we kick out some NADH, and we kick out some ATP, right? So we put in 2 ATPs here, and then we kick out 2 ATPs for each pyruvate down here. So 4 ATPs go out. And what they're saying, this diagram's in your text, they're saying we can actually slow down or control this hexokinase enzyme that prepares the glucose with a phosphate group to go through the process. And I also talk about that if we slow this step down, the glucose can move in and out of the cell membrane easy enough. But if we put this phosphate ion on it through this enzyme, then it doesn't easily move back through the cell membrane. So it's kind of trapped in the cell if this step goes through. So what they're saying here is if there's enough energy already in the cell and it doesn't need to continue to break down glucose, or there's already too much glucose in there, we can slow this step down and allow the glucose to just move from this cell out and go to the next cell. Maybe the next cell needs it. This cell doesn't need it. If this cell doesn't need to break down more glucose, we can inhibit this enzyme and prevent this step from occurring. But once this step occurs, then this guy's trapped and it has to go through the rest of the process, right? So maybe the cell wants to hold on to the glucose that it's got. It doesn't want to let the glucose go, but it's not ready to further process it. So we can actually inhibit this enzyme, preventing the this glucose, right? I guess this fructose here from getting a second phosphate group, because once you attach the second phosphate group, then it's easy to break it into 2 3-carbon chains. If we stop this step, then it's harder. We can't break this guy into 2 as easily. And once this guy's broken into 2, then it goes through the rest of this process. It generates NAD and NADH very easily. And then and then down at the very end, if we have plenty of ATP, we can actually slow this step down and not generate too much ATP within the cell. So quite a few different ways that we can regulate. Again, how do we do that? Well, these are all enzymes. See the ASE at the end of these names. These are different enzymes. So if we can slow those enzymes down through some sort of inhibition process, then we can slow this entire glycolysis process down and not have a buildup of too much NADH and too much ATP within the cell if we're not using it. Or again, also we can control whether glucose can easily move into or out of the cell. All right, so a little bit about regulation of respiration. We can control those enzymes as much as we need to. And that really kind of finishes up chapter 7. I'm sure I'm missing something there, but that should pretty well get us to the end of chapter 7. So I'm going to do just a few videos about chapter 8, and then we'll call it a day, okay? So I'm going to stop here because we're close enough to 5 minutes, and we'll call that good.